in 2012, my younger brother was diagnosed with brain cancer. And I decided to take what I knew about cell-cell communication and try to help develop new diagnostic tests for screening for cancers and different types of diseases so that we could catch them earlier. So I started really researching these small liquid droplets that cells make called exosomes in the hopes of using them for liquid biopsies, diagnostics, and screening for disease. And in the process of doing that, I realized very quickly that there wasn't a good way to get those exosomes out of the body fluids very effectively. It's like picking out peas out of your mother's vegetable soup, right? It takes a little time if you don't have the right tool for it. So it takes about 3.5 hours to do it with ultracentrifugation. And that is really time-consuming, labor-intensive, and would never work for quick liquid biopsy types of screening or diagnostics. So I worked with a partner in analytical chemistry here at Clemson University, and we developed a new way of isolating exosomes from body fluids in about 15 minutes. And we've developed a kit to do that.
We have little polypropylene columns that contain our proprietary capillary-channeled fiber for the capture of exosomes. We include little microfuge tubes into which you can capture your sample. And then we also include the buffers for eluting and cleaning your sample when it's on the column. All those things are in the kit.
Scientists have known about exosomes for many years, but they thought they were just a way of cells getting rid of extra material. It's just like if somebody commits a crime in their home and they throw out their trash, there's probably going to be some evidence in those trash bags. And so scientists really started looking at them again about 15 years ago, and they discovered very quickly that they're a treasure trove of information. They're like messages in bottles that cells, all cells in your body, send out. They put these messages, which may be RNA, maybe DNA, maybe proteins, in a lipid droplet. It's like a snapshot of what's going on in a cell in your body. So that is really why we decided to create Victory ExoFibres, is to basically put the power of this kit into the hands of scientists like myself who are working on very important diagnostics in the area of liquid biopsy. We are at the point where we have developed kits. We have done testing to make sure that they're translatable into a normal working molecular biology workflow in a lab. And our feedback is they love it.
South Carolina Research Authority, SCRA, has been extremely helpful and supportive. We've received one of their ASAP grants, which is Academic Startup Assistant Program, ASAP, which is designed to help companies take technology from university research labs into the marketplace.
They also work closely with a lot of the universities and the university tech transfer offices. Our tech transfer folks here are fantastic, and they are really connected with SCRA very closely, and that's been very helpful. SCRA has really come in and helped us to move from the bench to the actual getting things into customers' hands. So I'm a scientist. I don't know about how to go from working in this laboratory to sharing what I've discovered in a way that can help other scientists and build a business from. So SCRA, I think, really offers a lot of mentorship opportunities for somebody like myself.
SCRA has been extremely helpful, not only in helping us fund the commercialization of our technology and taking it out of the lab, bringing it into the market, but also with making introductions that will enable us to automate the manufacturing of our kits in the future.